Welcome people, today we're going to be discussing about um, glycolysis. And glycolysis, we're going to be discussing the process of cellular respiration. And this, in the process of glycolysis, there will be there will be two-step process, and then the definition. And we're going to be discussing, and I'm gonna be discussing about this type of process, and it's with simple words and as easy as possible. So let's get started. So, glycolysis, what is its definition? So, glycolysis, in simple words, is the breakdown of the glucose molecule, which in other words, a glucose is a type of sugar. It's a molecule that is in sugar molecules. So of the glucose molecule into 3 carbon molecules while producing ATP, adenine triphosphate, and then NADH, which is a nicotinamide adenine dinucleotide. So let me try to break down this explanation. So what glycolysis actual definition is, it means the breakdown of glucose molecules to producing more ATP molecules. So if we break down glucose molecules, we could produce more ATP molecules that could be used not just for that could be used in anaerobic respiration, which is a type of respiration used in conditions of no oxygen. So let's continue to the next page, and now at this coming slides, we're going to be discussing about the first half and the second half of glycolysis. Okay, so the first half of glycolysis, so the main topic question is, what occurs in the first half of glycolysis? So number one, this first step, it is hexokinase, a type of enzyme that catalyzes the phosphorylation of 6 carbon sugars. 6 carbon sugars are six type of sugars. 6 carbon sugars are bonded together. So, for example, glucose, 6-phosphate, a more reactive form of glucose. The reaction won't let the glucose from interacting GLUT. GLUT is a type of protein molecule, so not being able to leave the cell because of the negatively charged phosphate. So in simple words, what I'm trying to say in the first step is hexokinase, a type of enzyme, will catalyze the phosphorylation of 6 carbon sugars, which means that it will catalyze 6 carbon sugars, producing a more reactive form of glucose, a more working form of glucose, in other words. So this type of reaction is not going to let the glucose from interacting an enzyme called GLUT, a protein called GLUT, because of its negatively charged phosphates. So that's like the simple saying of the definition of the step 1. So let's get to step 2. Isomerase, a type of enzyme, will turn the 6-glucose phosphate into one of its isomers called fructose 6-phosphate. What this is trying to say is a isomerase, an enzyme called isomerase, will turn the previous 6-glucose phosphate or 6 sugar carbon into one of its isomers, which is called the fructose 6-phosphate. Alright, so it's the second step is more simpler than the first step because it's shorter. So let's just continue to the third step. In this step, the fructose 6-phosphate, which was from the second step, will be catalyzed, producing fructose 1,6-biphosphate. It is catalyzed by a high energy donation from the ATP molecule. So what I was trying to explain in this step is that the ATP molecule will donate a high energy to the fructose 6-phosphate. It will donate high energy to the fructose 6-phosphate, catalyzing and producing a fructose 1,6-biphosphate, which is a type of isomer. So let's go to the next step, which is the fourth step. And now it's gonna start to get more complicated, so I'll try to explain as simple as possible. An enzyme called aldolase is a type of enzyme, will break down the 1,6-biphosphate, which is a biphosphate from the third step that's produced into 3 carbon isomers. 3 carbon isomers, which is a dihydroxycetone phosphate and glyceraldehyde 3-phosphate. These are the 2 types of isomers that are produced from the breaking down to 1,6-biphosphate. So what this part is saying is that the 1,6-biphosphate, the 1,6, is broken down into 2 carbon isomers, which is dihydroxycetone and then the glyceraldehyde. So this thing is broken down to these 2 isomers. So now let's continue to the final step of the first half of glycolysis. So in this step, the isomerase transforms the dihydroxycetone phosphate into its isomer called glyceraldehyde 3-phosphate, which is this isomer. In the pathway, the breakdown of the glucose molecule leads to the energy of some molecules. So what this is trying to say is the breakdown of the glucose molecules leads to the net energy of sorry if you can see, this part means ATP. The thing that's covering is the ATP molecules. So let me write it down. The ATP molecules, which is a type that it's like a fuel. It's a type of like a fuel to cells, or in other words, it's the adenine triphosphate, which we discovered before. So what this sentence right over here is trying to say or convince is that the glucose molecule will be broken down into 2 ATP molecules. So yeah, so this is the first half of glycolysis. So first half glycolysis mainly, so what occurs in the first half, so we're gonna write down, it mainly occurs the formation the formation of 2 ATP molecules. So this is what mainly occurred in the first half of glycolysis. So the mainly event that happens is the formation of a single glucose, or the breakdown of a single glucose into two ATP molecules. So yeah, we're done with the first half of glycolysis. So what are the outcomes of glycolysis after we discussed about the first half and the second half, as we just discussed? The outcomes of glycolysis. We're going to discuss about what it's useful and what are the outcomes of this process. So it ends with two pyruvate molecules, 43 molecules, and two molecules of 8 ADH. So as we see, it will end up with two pyruvate molecules, which are produced in the last or the 10th step. 43 molecules, which is produced in the first step and at the last steps, and then two molecules of NADH. So that's the first part. The second outcome of this is that this is the part what's useful for. What's the usefulness of glycolysis? So the usefulness of glycolysis is that since mammal blood cells cannot convert energy, cannot produce energy, not being able to use aerobic respiration, which is the converting of energy with the presence of oxygen. They will use glycolysis in order to convert as their source of ATP production. Alright, so if I'm getting nervous, it's just that this is my first time putting this on YouTube, so I'm kind of getting nervous, but next time when I'm posting, I'll try to do my best, okay? Alright, so this is the sense, okay? We discuss about this part. We'll use glycolysis as a sort of ATP production. What this means is that since not everyone can use aerobic like we do, like breathing and stuff, they're gonna use anaerobic respiration, which is a type of respiration that converts energy without the use of oxygen. And that occurs with glycolysis. Glycolysis is the part of anaerobic respiration. That's the second outcome. The third outcome is, the third usefulness of glycolysis is that with damaged glycolysis, what happens? So this is the good usefulness. Good. Bad. So what happens when glycolysis is damaged? When glycolysis is damaged, cells will start to lose their ability on maintaining the sodium-potassium pumps, resulting in death. So if they start to get damaged, they're going to use your potassium sodium pumps, which is a type of pumps in the cells, resulting death. So here's the discussion about glycolysis. I hope you like it, and then see you next time when I create another video about these types of studies, and have a nice time with your studies. Put a comment how did you put a comment if you liked it or not, and yeah.